It's my pleasure to introduce our first speaker this afternoon, Dr. Feng Zhang. I'll keep the introduction for biographical things brief because you all have that in your book, but he is a core member of the Broad Institute of MIT and Harvard, as well as an investigator at the McGovern Institute for Brain Research at MIT and the W.M. Keck Career Development Professor at MIT, and he is a joint appointment in the departments of brain and cognitive sciences and biological engineering. So the focus of his research is to understand the molecular machinery of brain cells through development and application of innovative technologies. And he's going to talk to us today about one of those innovative technologies that he's been involved with. So he's very interested in how the brain works in neuroscience questions and has spent much time and effort and much productivity in developing technologies to help address those questions. And so those two technology that are so important in this, well, in all kinds of biology these days are optogenetics, which is not the focus of today, but of the genome editing mechanisms using CRISPR-Cas. And that's what we're going to hear from him today. So he's developed CRISPR-Cas systems to enable new, cheaper, and more effective ways to manipulate mammalian genomes. And his focus, as I've said, is on the brain and potential applications well beyond neuroscience. And he's been very, very helpful to the community in making reagents available and in tips and in helping people with the use of these new and exciting and very powerful technologies. He's the recipient of many prizes and awards, which I won't mention all of them. He received his AB in chemistry and physics from Harvard College, his PhD in chemistry from Stanford, and he's now holds the positions that I mentioned at the beginning. So the title of his talk is Development of CRISPR-Cas9 for Genome Editing. Feng.
Well, good afternoon. It's really a great honor to be here, and I want to thank John for inviting me, and it's really honor to be here. So yesterday afternoon, I had an opportunity to meet with some of the students here at University of Toronto, and I was really impressed with all of the students. And one of the things that they were very interested in is where I grew up. So I actually had made some slides for a different talk previously, so I just stuck in a couple of slides here, so maybe I'll take about 60 seconds just to show you a few things. Well, so I was born in the 1980s in China, and it was a time when China was just undergoing dramatic transformation. And so this is a slide, this is probably some of the things that you may see if you're walking around the streets in China. There's a strong emphasis on science and technology, and I grew up in that culture. So fortunately, my parents, my mother immigrated to the United States, and so in 1993, I was lucky enough to move from China all the way to the U.S. And there, I really owe a lot of my education and training. So China and the U.S. are very different. So here is what China looks like. There's billions of people, and this is what I saw when I landed in Iowa. I exaggerate a little bit. Des Moines is actually a fairly civilized place. There are fantastic school systems. And so I went to a public school called Colony Middle School. And this was really my first foray to molecular biology. There was a Saturday enrichment program, and one of them was on molecular biology. So I went, and they showed us a documentary, and this is a documentary. So from then on, my parents are computer programmers and computer engineers. And so growing up, I was always interested in how to program things. And Jurassic Park told me that biology is potentially another programmable system. And it made a huge impression on me. And also, my mentor from that class remembered it. So by the time I was a sophomore student in high school, he approached me and said, there's this fantastic opportunity at the Iowa Gene Therapy Center, Research Institute. And what about going there and trying to learn some molecular biology? And so I learned tremendous amount. John Levy was my mentor, very patient, sat down with me every afternoon just to teach me various things. He took out sheets of papers and can draw all sorts of fascinating biological processes. And many of those things I learned from then, I still use today, even when I show my graduate students how to do certain experiments. So following that, I went to Harvard and then had 2 additional really fantastic mentors. There's Don Wiley, a crystallographer, and also Xiaowei Zhang, who I worked with for 3 years to learn how to do biological imaging. So it's during undergrad, both through seeing my friends suffering from psychiatric disease, but also from me just wanting to understand more about my own mind, that I started to become interested in neuroscience. And that's when I wanted to focus my research on trying to understand brain processes and also try to help with neurological diseases. So I went to Stanford and then worked with Carl Dijsselroth on developing a technology called optogenetics. But following graduation, I wanted to do something that's a little broader. And so this is really the start of what I wanted to tell you about today. So here's a list of neurological and psychiatric diseases. What is common about this disease is that we don't understand the molecular mechanism, nor do we have very good treatments for most of them. So fortunately, the work of many pioneers have revealed that there are underlying and deeply rooted genetic and also likely epigenetic mechanisms for these neurological processes. And so if we can figure out a way to systematically unravel these mechanisms, then we may be able to develop a new suite of treatments to solve these diseases. And so one of the advances that we can really capitalize on is the advance in human genome sequencing. So here on the right is a plot from the National Institutes of Health, and it shows the number of genome-wide association studies that have been published over the past decade. And you can see there's an exponential growth trend, and each one of those data points, each one of those papers represents at least one or if not many more hypotheses that need to be tested. And indeed, these genetic variations imply correlations, but they don't imply causation for this disease. And so one of the things that's important for us to do is to be able to more rapidly and systematically go through and try to figure out what are the causal genetic mutations. So one way to do this is through the generation of models. And how do we go about generating models? Well, if you can generate a cell or animal model that carries the specific mutation that you want to study, and then compare it with the wild-type animal or cell, then you can do A/B testing and try to figure out if this specific variant really plays a causal role in the disease state. And we can do this fairly well in mice. There's gene targeting developed by Mario Capecchi and others. But to do this systematically at high throughput in any organism we want has been quite a bit of a challenge. So one way to solve this challenge is through the use of genome editing technologies. And so genome editing uses a tool called nucleases. And the idea is very simple, and it was really discovered by Jim Haber and also Maria Jason more than 2 decades ago. What they found is that if there is a double-strand break in a specific location in the genome, then this double-strand break can trigger endogenous DNA damage repair processes. There are 2 major pathways that we take advantage of. One is called non-homologous injoining, and this is an error-prone process. It's shown on the left side. When the 2 broken pieces of DNA are detected by the NHEJ machineries, they will get rejoined together, and oftentimes that will lead to the formation of a small insertion or deletion mutation. We call this the indel mutation. So if the indel is introduced in the coding region for a protein, this can lead to a frameshift mutation and allow us to knock out the gene function. This works very well, and we use this, and many other are now using this to generate knockout models. The more precise method and even more powerful is the HDR repair process, which has already been taken advantage of for generating knockout mouse ES cells and subsequently knockout mouse models. But here, if we can use a nuclease to generate a site-specific DNA double-strand break, this will allow us to increase the efficiency of HDR targeting by several orders of magnitude. And when that happens, we can provide a repair template and be able to switch in or remove specific sequences with much higher levels of efficiency. And so in order to realize the potential of genome editing, what needs to be developed are ways to be able to programmably introduce DNA double-strand breaks within a specific site in the genome. And so to do this, there have been many technologies developed, but they all hinge on the underlying principle, which is having a DNA binding domain that is programmable. And so here is one schematic of one implementation of this technology, whereby taking a DNA binding domain that you can easily alter the binding specificity of, you can consider this as a genome positioning system, a GPS. And you can use it to tether enzymatic domains, nucleases, transcription modulator domains to a specific location in the genome. And if you recruit a nuclease to the target site, then you can stimulate genome editing. So a few different DNA binding domains have been developed. One of the earlier ones is called zinc finger proteins, and a more recent one is called tails. And we just heard Ben talk about using talons for doing genome editing. But the two technologies are somewhat cumbersome to use because it's quite difficult to program a protein to be able to recognize a new DNA sequence. You have to either go through elaborate selection processes or to be able to construct together very repetitive sequences in a very systematic way. And so I started with zinc fingers and then later on worked on developing the talon system. But when I was working on it, I thought, this is not easy to use. Can we come up with something that's a lot simpler and will allow us to be able to target many more genomic regions with ease? And so there was other solutions. So nature is much smarter than us. And it turned out that over billions of years, bacterial cells have already developed a system to be able to achieve DNA targeting in a programmable way with significant ease. And this is the CRISPR system. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeat, which is a mouthful, but CRISPR is a lot crisper to say. And so basically, it's an adaptive immune system where when a virus infects the bacterial cell, there are small fragments of DNA sequences that are incorporated into that bacterial cell from previous infections that can get activated and transcribed into RNA to then guide enzymes from the CRISPR system to destroy the invading DNA. And so using RNA as a recognition motif to be able to recognize the invading DNA through base pairing, these enzymes can more or less target virtually any DNA sequence that you want to target. So this is a simple picture, and we often take for granted that this is really discovered by a large body of scientists working in many different countries. And in fact, this is just a brief summary of some of the major contributions to the field. It was first discovered in 1987, serendipitously, when scientists were looking into the genomes of E. coli and found these repetitive sequences. And then in 2005, a major finding that outlined the bold hypothesis that CRISPR is an adaptive RNA-guided immune system. And then over the subsequent years, it was demonstrated to be indeed an effective system in 2007. And then multiple groups worked out the components and also the molecular mechanisms of the CRISPR-Cas system. And so based on all these early pioneers' work, we harness the Cas9 system by introducing the Cas9 enzyme, as well as working out two different ways of delivering the RNA. One by either delivering the CRISPR RNA array, which consists of these target sequences along with the trace RNA, which was discovered by Emmanuel Charpentier, or by re-engineering a single guide RNA that was initially reported biochemically by Charpentier and Doudna, but then we figured out that you have to have additional sequences which would make it work in human cells. And so using this, you can then achieve RNA-guided DNA targeting and then be able to facilitate genome editing. So our work on this started, was inspired by a paper that was published by a Canadian researcher. His name is Sylvain Moineau. So back in 2011, when I was just starting my lab, I had heard about CRISPR through one of the meetings at the Broad Institute. And then when I went to look at CRISPR, I saw this paper that was published from his group. In this paper, he reported that the Cas9 system, back then it was called Cas5, is an RNA-guided DNA endonuclease. And so in this figure, he showed that the green highlighted sequence is the spacer from the CRISPR system, and that's a RNA sequence that when complements with the target DNA, either it's a plasmid or a phage, will facilitate a very precise targeted DNA double-strand break. And so that's where that break in the Sanger sequence trace shows. So I thought this is amazing. If you can use RNA to target DNA and to achieve site-specific DNA cleavage, this will be a lot easier to use than either zinc fingers or TALEN systems. And so there were many scientists working in the CRISPR field, and so just a few months later, Emmanuelle Charpentier, working in her lab in Sweden at the time, reported that in addition to the guide RNA and also the Cas9 enzyme, there's a third component of RNA that's required, which is the tracer RNA. And so tracer RNA complements with the CRISPR RNA by base pairing in the repeat sequence of the CRISPR RNA, and this dual RNA guides Cas9 in terms of targeting DNA and providing degradation function for infectious viruses. And so based on this early work, we thought maybe we can try to harness these systems and try to see if they can work within a eukaryotic cell to facilitate human genome editing. And so at the time, the field was working on trying to understand the CRISPR system, and some of the applications were using this to be able to improve industrial starter culture strains to be able to make yogurt cultures that don't get spoiled because it will get infected by phage viruses. But we thought maybe we can use it to edit genomes instead, to study genetic variations and potentially to be able to advance gene therapy. So I had to do a number of different things, and early on I wrote a few grants, but they unfortunately weren't funded, but one of them was funded, so this one was submitted back in January 2012, and basically we proposed to introduce the various components that Emmanuelle Charpentier described in her paper, namely Cas9, the CRISPR RNA, the tracer RNA, and also the bacterial host RNA3, which is thought to be important for RNA-guided, for processing of the CRISPR RNA. And so basically, we thought we can express these different components, the Cas9 enzyme, the CRISPR RNA, the tracer RNA, and RNA3, to see if they can reconstitute an active CRISPR complex within human cells. And so we started to work on characterizing each one of these components. And the first thing we did is to try to see if we can express Cas9 and also the RNA3 within human cells, and more importantly, to target them into the mammalian nucleus, which is something that bacterial cells don't have. And so on the top, we use nuclear localization signals to target Cas9 to specific to the nucleus of human cell. And we found that you have to have two. You have to really, Cas9 is a large protein, and you really have to flank it on both ends with the NLS to get efficient nuclear targeting. And also for RNA3, you have to have targeting or attaching the NLS on the C term in order to get it in effectively. So those are the two protein components. The second, the RNA component, we thought to see if they can be expressed in human cells. And so based on the RNA-seq data that Emmanuelle had published, she showed that there are multiple tracer RNA isoforms that are naturally expressed in bacterial cells. And so this is on the top here. We took each one of these isoforms and expressed in human cells, and what we found is that actually only one of the isoform was able to be expressed inside of a human cell. So on the bottom, you can see northern blot, where the top, the longer red bar indicates the full long natural isoform, but that one didn't express, and it's a shorter one that's also found in bacteria that is expressed within the human cell. So then we thought, let's see if this system can target the human genome. So we put in, we tried two different Cas9 molecules, one from S. pyogenes and another one from S. thermophilus. And then by altering the CRISPR array, by replacing that blue spacer with a piece of DNA sequence that corresponds to the EMX1 genomic sequence of a human cell, we were able to try to see if we can retarget Cas9 to cleave this piece of endogenous human genome. And what we found is very interesting. So on the right, this is a gel. We use an assay called Surveyor to detect modification in a specific locus in the human genome. And so the rightmost line shows introduction of all four pieces. So there's Cas9, there's RNA3, there's the CRISPR RNA, and also there's the tracer RNA. And then the second to last line shows three components without RNA3 being provided. And what we found is that you actually didn't need to provide RNA3 within a human cell to be able to achieve RNA-guided DNA targeting. And one nice thing about the CRISPR system is that it's a naturally multiplexable system. So these CRISPR arrays encode multiple spacers that can target multiple sequences, for example, multiple viruses. And we thought maybe we can use this to have CRISPR-Cas9 target multiple sequences within the human genome. And so what we did is we introduced three things. We put in Cas9, the tracer RNA that can safely express in a human cell, and also a single CRISPR array that carries both two spacers, one targeting the gene EMX1, and the second spacer targeting the neuronal interneuron marker parvalbumin. And what we found is that this system can work pretty well for targeting multiple genes using just a single piece of CRISPR RNA. And so when you have both of these spacers, you can detect robust modification in both of these gene targets. That's the first two lanes in the gel. And then if you only provide individual spacers, then you don't see modification in the second locus, suggesting that this indeed is a specific RNA-guided genome editing effect. So while we were working on this, there were also other groups working on studying the Cas9 system as well. And so Virgil Siksnes in Lithuania and also Charpentier, now collaborating with Jennifer Doudna, have published a very nice biochemical study showing that you can purify Cas9 and cleave DNA in vitro. And one thing that they did is they tried to figure out what is the minimum but sufficient component for RNA-guided DNA cleavage within a human cell, within a test tube. And so what they showed is that if you look at the cRNA and also the tracer RNA, the tracer RNA can be substantially truncated into just that very short fragment that's on the bottom, hybridizing with the cRNA. And so taking this information, then they show that you can actually link the two RNAs together to form what they call a chimeric RNA. And when they tested this in vitro to cleave DNA in a test tube, they found that whereas the last lane is the chimeric RNA, where you fuse the two pieces together, the second to last lane is the full-length tracer RNA. So that's not just the short blue fragment, but the whole thing, along with a cRNA that can target the target DNA sequence. And so it very convincingly demonstrated that the chimeric RNA can work even more robustly than if you put the two RNAs together in the cell. So we thought this is great. If you can simplify the RNAs to just two components, you can make the system even simpler. So we thought to compare the dual RNA system that we're working with the single chimeric RNA. And so this is targeting the EMX1 locus using the dual RNA, so with the CRISPR array and the full-length tracer RNA, so two RNA pieces. And this is what we found, which we were very surprised by, which is that the chimeric RNA with the components that work very robustly in vitro didn't have activity within the human cell. And so by comparing both of these RNA structures, we thought maybe the secondary structures on the end of the tracer RNA, which are not essential in vitro, might actually have important functions in vivo. And so based on that hypothesis, we started to lengthen the chimeric RNA. And what we found is that as soon as you started to include the first or both of the hairpin, then you can very dramatically rescue the activity. And so now, based on the crystallization work that we had done in collaboration with Osamu Niriki, we know that these secondary structures are actually important for interaction of the guide RNA with the Cas9 protein. And so just to sum it up, you can do genome editing using two forms of the RNA system. You can either put in Cas9 with the dual RNA system. One benefit of that is that you can encode multiple spacers into the CRISPR RNA array to be able to target multiple sequences at the same time. Or you can provide a single guide RNA with the full-length tracer RNA. So since then, we had adopted a three-pronged strategy to further develop the CRISPR-Cas system. So the first is to further advance CRISPR biology so that we can better understand the system and to be able to improve the specificity, the efficacy, and also various other properties of the technology. But at the same time, we wanted to make sure the technology is useful for a broad range of biological applications. And so we tried to work on developing high-throughput screening capabilities using the RNA-guided nucleases, rapid ways to be able to generate mouse models or even higher-level animal models, and also developing ways to be able to deliver this in vivo to study gene function in an efficient way. And then the third is to share the technology, make sure that it is open and widely accessible to really enable the community. So in addition to using Cas9 as a nuclease, there are also other ways of using the system. You can inactivate Cas9 to use it as a RNA-guided DNA binding domain to recruit effector domains to a specific locus in the genome. You can turn genes on or repress genes, or you can put GFP on it to be able to visualize things. But another thing that's very exciting is to be able to do genetic screens where by synthesizing 60,000 or 90,000 guide sequences, be able to very comprehensively perform loss of function or gain of function perturbation in the genome to identify new genes of interest for a given biological process. So we have been working on also trying to improve the Cas9 system, and one of the primary concerns is the targeting specificity. And so I'll show a little bit of unpublished data. So based on the crystal structure of Cas9, what we observed is that the inside the protein that interacts with the RNA and the DNA is highly positively charged, and that gave us the thought that maybe the way that Cas9 cleaves DNA is by using these positive charges to be able to stabilize the denatured DNA so that it doesn't rezip back up together and allow the RNA to invade. And so if this stabilization is too strong, then even if the RNA doesn't fully match, it can probably still hold the DNA open and allow Cas9 to make a cut. And so we thought maybe if we weaken these interactions by weakening these positive charges so that the DNA will more likely zip back together, we can try to improve the specificity. And so we generated a number of targeted mutations within this region, and what we found is that the effect is quite dramatic. And so on the bottom are some of the data from this study, where on the left side, the first set of bars show the wild-type Cas9 protein. And basically, if you look at mutations, so the two blue letters show mutations that are mismatches introduced along the length of the guide RNA. And what you see is that mutations that are farther away from the 3' end are fairly well tolerated by the wild-type Cas9 protein. But if you introduce these substitutions that neutralize the positive charge within this groove, for the enhanced SP-Cas9 1.0 and 1.1, you can see we more or less overcome these types of non-specific interactions for cleavage. And so this is a subset of the data, and what we are now working is trying to really understand what are the best combinations of residues that we can use to take this approach to improve Cas9. And I think an approach like this will allow us to really significantly improve the specificity and precision of genome editing. So for the rest of today's talk, I just want to share with you some of the most recent work that we have done on the discovery and harnessing of new enzymes for doing genome editing. Nature is amazing, and it contains many diverse organisms, and we thought there might be other powerful enzymes that we can harness to further advance and expand the genome editing toolbox. And so the first story is on the development of shorter, smaller, more compact Cas9s for in vivo delivery. And so David Cox is an MD PhD student in my lab, and he wrote a very nice review describing potential therapeutic applications of genome editing. And he summarized it as more or less just two approaches. One is ex vivo delivery, and one is in vivo delivery. So for diseases where the affected cells can be taken out of the body and repaired and then put back to the patient, we can do ex vivo delivery. So, and there are many different methodologies available for introducing enzymes or DNA vectors into cells in a petri dish. But that's only amenable to a small set of diseases, mostly things that affect the blood system. But for other diseases, we cannot take the organ out of the body. We can take the brain out of a person, fix it, and put it back. And for those, we may have to do in vivo delivery. And turns out that S-pyogenes Cas9, which is the most commonly used one, is very large, and it makes it challenging to package into a single adeno-associated virus vector. And so we thought maybe we can make something that's more compact. And so we had taken two previous efforts to compact Cas9, make it smaller. One of them is to look for, to work with this other Cas9 from Staphylococcus thermophilus, which is about 3.4 kb long. But it turned out that the activity level of this particular Cas9 is not as robust as S-pyogenes. And the second approach we took was then to use structure to guide the removal of excess domains that may not be critical for Cas9 function. But for that, we also found that we can get some success and reduce it by maybe 400 to 500 base pairs, but that's not small enough, and also it weakens the activity of the enzyme. And so we thought, well, this is not good enough. We really have to come up with a way that's more efficient, and maybe we can seek inspiration from nature again. And so one thing we did is we looked at all of the Cas9s that exist in nature in sequence databases, and then we plotted their length along the x-axis. And so what you see here is that Cas9s fall into two length distributions. There's the smaller cluster that centers around 1,000 amino acids, and then there's the longer cluster that centers around 1,350 amino acids. And so S-pyogenes Cas9 belongs to the longer cluster, the 1,350 amino acids, and we thought maybe we can harness the smaller Cas9 that would work well in human cells from the shorter cluster. And so what we did is we did a phylogenetic reconstruction of the shorter cluster, and we picked 6 different short Cas9s that kind of well represent this, the diversity in this small cluster. So what we found is that, So what we found is that if you test the activity of these small enzymes in vitro by expressing them in human cells and then lysing the human cell to take the cell lysate, all of the small Cas9s are functional within human within the human cell lysate, and we can figure out what the PAM sequences are, and then we can see pretty robust DNA cleavage. But when we actually tested genome editing in human cells, in living cells, we found that only one of the 6 Cas9s worked, and that is a Cas9 from Staphylococcus aureus, and that's the third lane that you see there. So S. aureus had comparable activity as both the pyogenes and also the Thermophilus Cas9, but the other 5 didn't have the same level of activity. And so that allowed us to then package Cas9 along with the guide RNA into a single adeno-associated virus. We focus on AAV because it's the only viral vector that has been clinically proven and is in a drug called Glybera in Europe. And so we thought, let's see if this can allow us to achieve efficient in vivo genome editing. And so what we did is we designed this guide RNA to target PCSK9, which is a prominent drug target for treating hypercholesterolemia. And so we thought, let's put this in a mouse and see if we can knock out PCSK9 using SA-Cas9 and then observe a phenotype. And so this is the data that we got. So on the first plot on the left side shows detection of indel formation in the PCSK9 gene in the liver of the mouse. And what we found is that the effect is quite robust. So within 7 days of AAV administration, we observed over 40% indel formation within the liver tissue. And then this modification efficiency more or less saturated, and it persisted for the remaining time points that we took. And if you take serum from the mouse to check both PCSK9 protein levels, which is the middle plot, or check the cholesterol level, which is the third plot, what we see is that within 7 days of injection, there's also a sharp drop both in the protein level as well as in the RNA, both in the protein level as well as in the cholesterol level. And so this showed that indeed we can build viral vectors that are compact and can mediate efficient in vivo delivery and achieve therapeutically relevant editing efficiency. So that's to develop a smaller Cas9, but there are other things that would be great to also address. For example, Cas9 generates double-strand breaks and is very good at knocking genes out, but for inserting genes, especially in post-mitotic cells where the HDR process is not very active, we really need to try to see if there are other possibilities. And so we asked, are there other enzymes out there other than Cas9 that can also provide genome editing functions, maybe with expanded functionalities? So if you look back to the classification of the CRISPR system, the latest one that was published by Eugene Koonen's lab reclassified CRISPR system into 2 different classes. There's class 1, which are the previously referred to type 1 and type 3 systems, and they use multiple proteins to recognize the guide RNA and then also to provide the DNA or RNA targeting and destruction function. Class 2 is the simpler one where Cas9 belongs to. It uses a single enzyme to recognize and destroy invading DNA. And we thought if there are multiple types of class 1 systems, there must be multiple types of class 2 systems as well. And so it turns out there are. And so working with Eugene Koonin, we examined one of these alternative class 2 systems. And this is a protein called CPF1. So it turns out that in the bacteria Francisella novicida, there are 2 CRISPR loci, one that contains Cas9 and another one that contains CPF1. And if you look at the domain organization comparison between these 2 proteins, you can see that Cas9 contains 2 nuclease domains. There's the H&H domain and the RUBC domain. RUBC is split into 3 different pieces that come together when the protein is folded. CPF1 contains a single known domain, RUBC, that's contiguous at the C term of the protein. And so we thought, let's see if this system is also active and if it's also a real CRISPR system. So after characterizing the biological function, we found that it indeed is an RNA-guided DNA endonuclease. And there are 3 differences between that and Cas9. The first is that the target sequence is that the guide RNA molecule is almost like the mirror image of the Cas9 CRISPR RNA. For the target site, the PAM sequence is on the left side, and for Cas9, the PAM sequence is on the right side. CPF1 uses AT-rich PAMs, and Cas9 uses GC-rich PAMs. The second difference is that CPF1 is a single RNA-guided endonuclease. It just uses a single short cRNA to provide the recognition, whereas Cas9 is a dual RNA-guided system. You have to have the CRISPR RNA as well as the tracer RNA. And then the third is that CPF1 cleaves DNA in a staggered fashion. It generates the sticky end, very much like a restriction endonuclease, HinD3, BAMH1, CLA1, so forth. Cas9 generates a blunt end DNA cleavage. And so this particular difference may be advantageous for trying to achieve gene insertion in postmitotic cells because the NHEJ process is very active in neurons. And if we can provide provide donor sequences that carry compatible overhangs, then we can potentially use NHEJ to insert sequences in a postmitotic cell. So we're very excited, and we thought to see, can CPF1 mediate genome editing efficiently? We tested Francisella CPF1, but we found that we were very disappointed. It didn't show activity in a human cell. But based on what we learned from Cas9, we thought, well, maybe FN CPF1 couldn't work, but there may be other CPF1s that can work. So we went ahead and we did a phylogenetic reconstruction, and then we picked 15 additional CPF1s that captured the entire diversity of CPF1. And then we tested them in human cells. So these are the 16, so FN plus the 15 additional ones. You can see the sequences are actually quite diverse. So when we're testing human cells, we found that even though all these CPF1s can work in vitro, so that's the gel, the human cell lysate gel that you see. Every single one cleave DNA robustly. But when you test it in vivo genome editing, only 2 of the orthologs worked. One from Staphylococcus and the second one from Lactospiraceae. But nevertheless, this shows that distinct bacterial species may have different environments that would render translation difficult, and it's important to find bacteria that have compatible intracellular environments that would permit easy harnessing into human cells. So we also compared the CPF1 efficiency with Cas9, and so we targeted 4 different loci, and we found that they have comparable levels of genome editing efficiency. So this is CPF1 and Cas9, and there are now 2 different enzymes, but are there additional enzymes? We thought maybe we can carry out a search to see, are there additional CRISPR effectors that can also be useful for extending the genome editing toolbox? And so working with Eugene Koonin and also Konstantin Svirinov at Skotech, we set up a computational algorithm to really mine through all of the genomic sequences by looking for any unknown large gene that's next to the CRISPR adaptation protein Cas1. And based on this, we found some new ones. So here are the 2 known ones, Cas9 and CPF1. And then there are 3 new ones that we just recently reported. There's C2C1, C2C2, and C2C3. C2C1 and C2C3 are more similar in terms of domain organization as CPF1, but C2C1 is a dual RNA-guided nuclease, whereas CPF1 is a single RNA-guided nuclease. And C2C2 is the most interesting one because it has a very distinct organization. So there's going to be many more to come. So we're continuing along this three-pronged development strategy, but what we're most excited about is that we're now ready to apply this to study nervous system function and also mutations in diseases. And so one of the things that we're working on is to be able to study mutations that underlie neurological and psychiatric disease. And so one is in vitro modeling by using stem cells and putting in mutations to study whether or not they cause specific phenotypes within the derived neurons. And the second is to generate mouse models of the same mutation and make comparative studies to better understand the function of the gene within intact circuits and also within the behavior of the animal. And so here are just two very quick examples. So about 3 years ago, it was reported that CHD8 is a novel de novo mutation that's highly linked to autism spectrum disorder. And so by using genome editing to introduce this CHD8 loss of function mutation into stem cells, we were able to derive neurons and then carry out electrophysiological characterization. And so what you see on the bottom left, the decreased evoked current response plot is a comparison of the firing threshold of these cells with respect to input electrical current. So what we found is that the knockout CHD8 cells have a more immature-like firing pattern. And then we can also do the same in vivo. So we also build mouse models that knock out CHD8. And by doing the social interaction test that Ben showed earlier, we found that there's also altered levels of social interaction within these animals. So these are just things that we're working on now that we're really excited about and that will hopefully help us better understand disease. But in the long run, what would be really exciting is to really refine the genome editing toolbox so that we can treat this long list of devastating genetic disorders. And we're working hard to make it possible so that it's efficient, specific, and also to be able to deliver it to the relevant tissue. So this is the final, most important slide. I just want to take 30 seconds. I want to first acknowledge all of the students and postdocs in my lab that are both passionate and also extremely creative in solving this problem, but also our wonderful collaborators, Luciano, who we worked with fairly early on the Cas9 system, Osamu to solve crystal structures, Eugene, Constantine to look at various new enzymes, and many other collaborators on applications, and then also many of my mentors who played a critical role in helping me learn how to do science and to be able to work effectively as a scientist, but also all of the funding agencies that supported us early on for these kinds of high-risk, potentially no payoff type of project. So thank you so much.
We will take 2 questions. Okay, thank you very much for this wonderful tour through this exciting technology. We've got time for 2 questions. We'll leave major questions for the round table after, but if there's a couple of questions where Dr. Zhang is happy to.
Are you speaking next?
Nobody? Oh.
Check.
Yeah.
Others probably can't, though. Let's get the microphone.
Thank you. One question I had was, I know in the review paper published by Dr. Cox, I believe he mentioned there's research going into potentially immune reactions because it is microbial derived. I don't know if you could comment on that a little bit.
Yeah, that's actually one of the most important challenges facing clinical translation. These Cas9 or CPF1 enzymes are microbially derived, and we haven't done enough characterization to understand the immune reactivity to the protein. But for long-term applications, that is certainly something that needs to be addressed. Maybe first taking fragments of protein and figuring out what are the most antigenic epitopes, and then trying to optimize them so that they don't evoke strong immune responses.